So if we do presentation, then we have to do it in group. And assignment, I think hopefully I will let you know about the topic assignment, and assignment will be individual work. Or maybe okay, let me think about it little more. I hopefully let you know the next class. Okay, so we were studying TCA cycle. Let me share the slide with you. Can you see it?
Yes, ma'am.
So আমরা এখানে ছিলাম না, cycle, TCA cycle. So we have already seen the glucose is catabolized. At first, it is catabolized by the glycolysis pathway, and we get the 3-carbon compound pyruvate. And pyruvate is further undergoes and in the next step, the acetyl CoA enter the TCA cycle. Okay, so this cycle, TCA cycle, is common for all the nutrients, and all the nutrients, whether it is glucose, amino acid, or fatty acid, all of them will be actually oxidized into acetyl CoA, which will enter the TCA cycle. Okay, now in the cycle we can see, because this is a cyclic metabolic pathway, and in a cyclic metabolic pathway, the starting material is always regenerated. So here acetyl CoA, which is entering the cycle, and it is condensed with oxaloacetate by the enzyme citrate synthase, and that reaction is known as condensation reaction, and we get 6-carbon compound citrate, because oxaloacetate is 4-carbon compound and acetyl CoA has 2 carbon, and they condense together and give rise to the citrate. And citrate actually sequentially undergoes dehydration and hydration, and like get the compound isocitrate. Isocitrate also has the 6 carbons. Now isocitrate undergoes oxidative decarboxylation, that means oxidation happens as well as decarboxylation, which is the removal of carbon dioxide. And this reaction is important, and this reaction is catalyzed by the enzyme isocitrate dehydrogenase, and because you can see the enzyme dehydrogenase, so that's why there will be the participation of the coenzyme, and in this case it is NAD plus, which will be accepting the electrons coming from the reaction and becomes NADH, and the carbon dioxide is released. Next step, we get the intermediate alpha ketoglutarate having the remaining carbons, and alpha ketoglutarate then undergoes another oxidative decarboxylation, and the enzyme is alpha ketoglutarate dehydrogenase complex. So that means this enzyme is not a single enzyme. Here multiple enzymes are working like the pyruvate dehydrogenase complex we learned, and we get succinyl CoA because of the participation of the coenzyme A, which will be added to the substrate, and we get the product succinyl CoA. Again, because of the dehydrogenase enzyme, and it is an oxidative reaction, we get NADH. Next step, succinyl CoA undergoes the reaction This is also very important reaction in this pathway, that is the substrate level phosphorylation. And we actually learned this reaction in the glycolysis pathway as well, where a substrate is phosphorylated. And here the substrate is GDP or ATP. So they are phosphorylated into GTP and ATP, and at the same time, succinyl CoA is becoming succinate by the release of the coenzyme by the enzyme succinyl CoA synthetase. And succinate is 4-carbon compound. Succinate is then undergoes dehydrogenation, and in this reaction another electron carrier FADH2 is produced by the enzyme succinate dehydrogenase, and we get the um, intermediate, which is fumarate, because here the electrons are released. That's why the double bonds in the molecule is produced, fumarate. And fumarate then undergoes hydration reaction, which actually removes the double bond, and we get the intermediate malate by the enzyme fumarase. And here the malate in the next step undergoes another dehydrogenation reaction by the enzyme malate dehydrogenase, which eventually regenerate the starting material of this cyclic pathway, that is oxaloacetate. So that is the cyclic TCA cycle or Krebs cycle or citric acid cycle or carboxylic acid cycle. It has many names. And here is the description of this cycle. But if we just like memorize the description, it will be very difficult for us to understand. Try to understand the reaction here, what is happening in the reaction, what type of reaction, and what are the enzymes involved in the reaction. So you can see the enzymes are different from each other. Some of the enzymes are called synthase, whereas others are synthetase, some are dehydrogenase. Okay, so you have to understand that, otherwise it will be very difficult for you if you memorize the description of the pathway. Okay, so next slide is about the enzymes. So this will help you to understand the various enzymes we are seeing in the different metabolic pathways. The first one is the synthesis, which we have already seen in cyclic TCA cycle. That is the acetyl CoA, sorry, uh, citrate synthase. Yes, citrate synthase. So these enzymes, they actually do the condensation reaction. That means they condense or combine two molecules, give rise to a single one. And these enzymes actually, they don't need energy. That means such as in the form of ATP or GTP, or we call that nucleoside triphosphate, but they can do the reaction by themselves. Another one, synthetase. Synthetase is also in the cycle. If you go back and check it here, succinyl CoA synthetase. Okay, so this is another enzyme. These synthetases, they also do condensation reaction, but they need ATP. That means they need energy in the form of nucleoside triphosphate. And the particular example here, that is the succinyl CoA synthetase. And next comes ligases. Ligases actually again doing the condensation reaction. That means they are actually Latin, and this ligase, it comes from the Latin word liger, which means you are tying together. And in this reaction, when the two atoms are joined, again this reaction requires ATP or energy. And example of ligases, if we say in this regard, because they have same way of function like the synthetases, so we can say synthetases are ligases. And another best example of ligase is DNA ligase, which is a very important enzyme in genetic engineering that is used to join DNA fragments together. Then comes the lysis. Lys is actually just opposite of the condensation reaction. because lysis is catalyzing the cleavage reaction. And here that happens by electronic rearrangements. And the best example is the pyruvate dehydrogenase complex, if you remember, which catalyzes the reaction where pyruvate is converted into uh, acetyl CoA and carbon dioxide. So this is the best example of the enzyme lyase. That means pyruvate is degraded and we get the acetyl CoA and carbon dioxide. Then comes the another very commonly used enzyme in metabolism, obviously, is called kinase. And kinase are the enzymes which are actually responsible for addition of a phosphate group to its substrate. And the source of the phosphate group is usually nucleoside triphosphate, such as we can think of ATP. And to the substrate here, the substrate can be actually different types of substrates, such as a sugar. And for sugar we already learned the example, such as hexokinase or glucokinase. Or maybe it add the phosphate group to a protein, and example of the protein is glycogen phosphorylase kinase. Then another nucleotide, example is nucleoside diphosphate kinase. And a metabolic intermediate such as oxaloacetate, example of the enzyme is phosphoenol pyruvate carboxykinase. So there are various kinases according to their substrate where they are adding the phosphate group, but the source of the phosphate group is usually a nucleoside triphosphate, example ATP. Next reaction is the phosphorylases reaction, where actually a compound, again we can say a compound is broken down, but that breaking happened by a phosphate group. The phosphate group is actually attract that particular bond, and then bond is broken. And the enzyme is called phosphorylases. And we have already learned one such enzyme, which is glycogen phosphorylase. And glycogen phosphorylase actually attack the glycogen, which is a polysaccharide. And where it attack the especially the um, uh, reducing end of the sorry, non-reducing end of the polysaccharide. And here when the glycogen will be degraded, it produces sucrose 1 phosphate. That means two sugar residues, or two sugar residues are attacked, and by that it will be leaving the polysaccharide as sucrose 1 phosphate. Or sometimes it can also produce glucose 1 phosphate. And the last enzyme in this list is the enzyme which is responsible for removal of the phosphate group or phosphoryl group from the substrate. And the substrate is here called the phosphate ester, and the enzyme is called phosphatases. So in this reaction, water is required. And example here we can see the fructose bisphosphatase 1, that is the enzyme fructose bisphosphatase 1, and its substrate is fructose 1,6 bisphosphate, because the enzyme is targeting the phosphate group which is attached to the number 1 carbon. So that will be released, and so we get the product fructose 6 phosphate. And this reaction occurs in gluconeogenesis. Okay, so these are the some enzymes which are involved in different metabolic pathways. And actually some examples we have seen it, but we can correlate with the examples we are learning in different pathways of metabolism. Is it clear?
Yes, ma'am.
Okay, so next again look at the TCA cycle. So what are the products of one turn of TCA cycle? So we can see that the major like results of the TCA cycle or production of the TCA cycles are here. You can see three molecules of NADH, and then one molecule of FADH2, one molecule of GTP or ATP, and two carbon dioxides are produced in one turn of the TCA cycle. And next one. So these TCA cycles is the one which is actually occurring in the living organisms, usually which are living in oxidative environment. But there are some organisms, specifically anaerobic microorganisms. So they have TCA cycle, but that is different from the complete TCA cycle. And that TCA cycle, it has different purpose. And the purpose is that they actually use that TCA cycle to produce the biosynthetic precursors. Okay, so if you look at this cycle, where this is not the particular cyclic pathway, but here the starting material oxaloacetate, it can go in two ways. It can be condensed with acetyl CoA to make the citrate, and like the TCA cycle, the citrate is converted into isocitrate. Isocitrate then is converted to alpha ketoglutarate. And in the another way, another direction, oxaloacetate is actually converted to malate, fumarate, succinate, and succinyl CoA. So oxaloacetate, that means here in this reaction, in this cyclic pathway, oxaloacetate is not regenerated. So the cell has to synthesize oxaloacetate to keep the cycle operating. So the source of the oxaloacetate in these anaerobic microbes is from the phosphoenol pyruvate or pyruvate. So these two molecule, they undergoes carboxylation reaction, that means addition of carbon dioxide and oxaloacetate is produced, which then actually running this particular incomplete TCA cycle. And what is the purpose? As I said, that the purpose is to provide the biosynthetic precursors. And what are the biosynthetic precursors here produced? One is the alpha ketoglutarate, and another one is succinyl CoA. So these are the four carbon compounds which are actually synthesized by this incomplete TCA cycle, and these precursors can be used to synthesize other biomolecules such as amino acids, nucleotides, heme, etc. Okay, so that is their main function in anaerobic microorganism. So they are not utilizing the cycle to generate energy, that means the reducing power, as we have seen in case of normal TCA cycle. Rather than they are using it to synthesize the biosynthetic precursors. So another important differentiation in between these two, in the previous one where we have seen already that the starting material always regenerated after each cycle, but here the no regeneration of the starting material. So obviously the cell needs to synthesize the starting material so that the cycle can go on. Is it clear?
Yes, ma'am.
Okay, now we will go back to the original TCA cycle. So here um, we are looking, or we are actually looking at the different intermediates of the TCA cycle, and actually these intermediates, they also have same function as the TCA cycle, incomplete TCA cycle happened in anaerobic microorganism, because that cycle's purpose is to produce biosynthetic precursors. And here in the original TCA cycle, actually the intermediates, they can also be used as biosynthetic precursors. So if you look at this cycle, so oxaloacetate, acetyl-CoA, they condense, we get citrate, then alpha-ketoglutarate, succinyl-CoA, malate. So these are the important intermediates actually which are utilized as biosynthetic precursors, such as from citrate, fatty acids and sterols are synthesized. From alpha-ketoglutarate, you can see the amino acid glutamate is synthesized, and from glutamate other amino acids as well as the base purines are synthesized. From succinyl CoA, porphyrins and heme group are synthesized. Then oxaloacetate, from oxaloacetate the amino acids aspartate, asparagines, they are synthesized, and from that pyrimidines, the base, are synthesized. So that means in this case, the cell is utilizing TCA cycle not only to produce the energy specifically, because the main function here to produce the reducing power NADH and FADH2 as well as ATP or GTP. That is the main function. But here we are seeing the TCA cycle is not only involved in that, it is also actually providing the precursors, because the intermediates, they are functioning as the precursors to synthesize other important biomolecules. So we can say that in aerobic organism, the TCA cycle is actually an amphibolic pathway. That means it has both features of anabolic process and catabolic process. When we are talking about catabolic process, that actually give rise to the production of the NADH, FADH2, GTP. And when we are talking about anabolic process, that is about the synthesis of other biomolecules from the intermediates of this cycle. Is it clear? Do you understand?
Yes, ma'am.
So the blue, actually here we are talking about the blue arrows. Please follow the blue arrows, not the red arrows. We did not discuss about the red arrows yet, that will come later. So if we follow the blue arrows, then we can see from these intermediates what are the important molecules are synthesized. Even you can see oxaloacetate, which actually used to synthesize other molecules, especially different amino acids as well as glucose. Okay, glucose. So oxalic acid is used to synthesize glucose. So that is very important. So just for the anabolic pathway, we have to follow the red arrows, where we can clearly see the intermediates are like the source to synthesize all the various biomolecules. And the original catabolic pathway that we have already learned. So this is the catabolic pathway, that's the outcome of the catabolic pathway, where we get the NADH, FADH2, GTP as well as carbon dioxide is produced. Okay, now the thing is that the question here can arise, if the intermediates of the TCA cycle are used as the precursors to synthesize other biomolecules, then what will happen to the TCA cycle? Can it actually operate? What do you think? If you using intermediates of this cycle to synthesize other biomolecules such as glucose, amino acids, or hello, what is your like um, understanding if a cycle is going on and you are utilizing the intermediates for another purpose? What will happen to that original cycle? Will it go on?
ম্যাম এটা কি তাহলে ইনকমপ্লিট
No
থেকে
ma'am,
যাবে?
incomplete থেকে যাবে এটা যদি আমরা ইন্টারমিডিয়েটস গুলো নিয়ে নিই মানে অন্য কাজে।
Yes, we cannot say incomplete. That means the cycle will not go on. সাইকেল তো চলবে না যদি ইন্টারমিডিয়েটই না থাকে, তাই না? তাহলে এখন সেলকে কি করতে হবে? Cell has to ensure that the intermediates are there. So cell has to supply the intermediates of the TCA cycle. এখন সেল সেটা কিভাবে করবে সেটাই হচ্ছে কথা, এটাতে আমরা আবার ফিরে আসব। Okay, now these intermediates which are removed from the TCA cycle, which are actually functioning as the precursors for some biomolecules, they are replenished. Okay, they are replenished, মানে তোমাকে রিপ্লেনিশ করতে হবে, তোমাকে সেই ইন্টারমিডিয়েট গুলো আবার এনে ওখানে রিপ্লেস করতে হবে, যেহেতু শেষ হয়ে গিয়েছে আবার তুমি ওখানে ফিল আপ করবা by reactions, by some reactions which are called anapleretic reactions. Okay, so the intermediates of the TCA cycle are replenished by some reactions which are called anapleretic reactions. So this table in this slide, this table is showing the common anapleretic reactions. Actually, those reactions are also shown in this slide. Now follow the red arrow. If you follow the red arrow, you will find the anapleretic reactions. So the major anapleretic, the major compound in this pathway is obviously the oxaloacetate. If the oxaloacetate is not there, the cycle cannot start. Okay, once there is enough oxaloacetate, the cycle will go on. So the cell makes sure that there will be enough oxaloacetate even after the use of the intermediates as biosynthetic precursor. So the cell is synthesizing oxaloacetate from pyruvate. If you look at this, pyruvate by enzyme pyruvate carboxylase, or from phosphoenol pyruvate by phosphoenol pyruvate carboxykinase, or another enzyme phosphoenol pyruvate carboxylase. Okay, and there is one more intermediate is produced, that is from again pyruvate by malic enzyme, which produce the intermediate malate. So in the next reaction, malate will be converted into oxaloacetate by malate dehydrogenase. Okay, so these 1, 2, 3, 4 red arrows, those are the reactions by which the cells actually replenish the intermediates of the TCA cycle, so that the TCA cycle must go on, otherwise the cell will be unable to produce the energy. So here, if you now look at the table of the reaction, which are the most common anapleretic reaction, if you look at here, you can see pyruvate to oxaloacetate, phosphoenol pyruvate to oxaloacetate, phosphoenol pyruvate to oxaloacetate, and they are done by the different enzymes which is mentioned in the figure as well. Okay, then pyruvate to malate. Actually, there are different reactions because those reactions are happening in different tissues or even different organisms. So liver, kidney, heart, skeletal muscle, then pyruvate to oxaloacetate, which is catalyzed by the phosphoenol pyruvate carboxylase, which happens in plants, higher plants, yeast, and bacteria. And the pyruvate to malate, that reaction occurs in eukaryotes and bacteria. That means both eukaryote and prokaryote. Okay, so by these, the cell is replenishing the intermediates, otherwise the TCA cycle cannot proceed. Clear?
Yes, miss.
Okay, so next one is the regulation of the TCA cycle. So as we have seen in case of glycolysis, we have learned about several factors which can regulate a metabolic pathway. So in case of TCA cycle, there are three factors that is responsible in the regulation of this cycle, and those are: number one, substrate availability. So you can correlate with the factors you learn from the glycolysis. Substrate availability, that means if there is no substrate, then the cycle will not go on. Then inhibition by accumulating products, that means if there are more products, then there is no need of the cycle operation. And then there is the allosteric feedback inhibition of the enzymes, the allosteric regulation of the enzyme. And these enzymes usually involve in the early steps of the cycle. Okay, so feedback inhibition always actually, which is done by the products, specifically end products, and they actually control the preliminary or the beginning pathways of the metabolic pathway. Okay, now, so here substrate is what? Substrate is acetyl CoA. If there is no acetyl CoA, or we can also think about the substrate is what? The because ultimately the goal of this cycle is the production of energy. So that means if there is more NAD and ADP, then the cycle will go on. But if there is enough product, which is like the our goal is the ATP and NADH, then the cycle will be inhibited. And the enzymes here, which actually involve in the early steps, actually the three steps in early three steps in the cycle, and they are what? Citrate synthase, which catalyzes the very first step, condensation of oxaloacetate with acetyl CoA. Then isocitrate dehydrogenase, that is the first reaction where NADH is produced. And third one is the alpha ketoglutarate dehydrogenase. So if you look at this, that means these enzymes, because they are directly involved with the production of NADH. So if there is enough NADH, that will inhibit these enzymes. Or if there is less of NADH, or maybe many substrates, NAD plus and ADP are present, then these enzymes will do the reaction. Okay, so that is the regulation of the TCA cycle, the major points actually. But if you look at this diagram here, many things are shown, but actually all of them are related to these three factors, if you can relate it. So first one, if you look at this, the cross sign obviously, which are the inhibitory factors: ATP, acetyl CoA, NADH, fatty acids. Because fatty acids, when there are many fatty acids which are oxidized, there will be more acetyl CoA, as well as NADH will be produced. And the like stimulatory factors are here, which are shown by the green arrow: AMP, CoA, NAD plus obviously, and there are some ions are shown, and those are actually most of the ions which are functioning as the prosthetic group for the enzymes. Then again, in the very first step, if you look at this, the inhibitory factors are NADH, succinyl CoA, citrate, ATP. So these are the like ultimate products of this reaction. And stimulatory factor is ADP. If there is enough ADP, the reaction will go on. Same way in each of the main step, if you look at this isocitrate to glutarate, again ATP, and the stimulatory one specifically ADP. Then next one alpha ketoglutarate. So if there is enough succinyl CoA, then this reaction will be inhibited, as well as enough NADH that will inhibit the reaction. Okay, so the major factors, if you show that will be enough to discuss the regulation of TCA cycle. Can you hear me?
No, ma'am.
No, ma'am.
Why you cannot hear me? Where are you?
এখন শোনা
ম্যাম
যাচ্ছে।
এখন শোনা যাচ্ছে ম্যাম।
So let me know where are we? Bolo. Where are we? What are we discussing?
ম্যাম রেগুলেশন অফ দ্য
Instead of TCA.
So if you are not listening, then why don't you let me know that you are not listening to me? Why?
কথা বেঁধে বেঁধে আসছিল।
So then you should have let me know there is problem. If you don't let me know, how can I come to know whether you are listening clearly or not? So let me, what do you understand by the regulation of the TCA cycle? Tell me. বলো TCA cycle regulation কি বুঝলা?
তিনটা ফ্যাক্টর দ্বারা রেগুলেট হয়। এখানে বলছে যে সাবস্ট্রেটের অ্যাভেলেবিলিটি, আর যদি এনাফ প্রোডাক্ট অ্যাকুমুলেট হয়ে যায়, আর অ্যালোস্টেরিক ফিডব্যাক ইনহিবিটেশন।
তো সাবস্ট্রেট এই ক্ষেত্রে কি? What are the substrates of TCA cycle? তো সাবস্ট্রেট আমরা কি বুঝতে পারছি এখানে? What are the substrates?
ATP, NADH.
ATP and NADH, are they substrate or product?
Product means substrate, pyruvate, acetyl CoA means.
Yes, acetyl CoA as well as NAD plus and ADP or GDP. They are the substrate, because the ultimate products are the ATP and NADH. তাই না? এখানে আমরা যেটা দেখতে পাচ্ছি, আমরা কি পাচ্ছি? Production of NADH and then production of GTP or ATP. So those are the final products. Okay, so those are the substrates. And then products are ATP and NADH. If enough ATP and NADH in the cell, so cell then will actually control the TCA cycle. And another one said about the enzymes, and those enzymes which are involved in the first or preliminary steps of the pathway, and they are actually regulated by allosteric feedback mechanism, where there are some end products which can actually inhibit the enzyme by allosteric modification. Okay, so that is the regulation of TCA cycle. Next one, that is about another cycle which is called glyoxylate cycle. And at the beginning we have, when we were learning gluconeogenesis pathway, if you remember, Gluconeogenesis is the synthesis of carbohydrate from non-carbohydrate sources. And we have also learned that vertebrates, they are unable to synthesize carbohydrate from fatty acids or the acetate. So why it is difficult for vertebrates? Because when a phosphoenol pyruvate is converted to pyruvate, or the pyruvate to acetyl CoA, because if you remember specifically, because we have learned this reaction pyruvate to acetyl CoA, that is irreversible reaction. Okay, and so much energy is required, and it is not possible to reverse the reaction. So this cannot happen in vertebrate. So that's why vertebrate cannot synthesize carbohydrate from fatty acid or acetate. Okay, now there is a reaction which also we learned in the TCA cycle, or specifically we can say where the cells actually generate the oxaloacetate to replenish oxaloacetate. So this, in this reaction, phosphoenol pyruvate, which actually can be synthesized from that oxaloacetate. So actually if we go back to the cycle, you can see that here oxaloacetate, we can see the red lines, red arrows, if you follow phosphoenol pyruvate to oxaloacetate, which is catalyzed by phosphoenol pyruvate carboxykinase. Okay, but at the same time, that means this reaction is a reversible reaction. So it is possible to synthesize phosphoenol pyruvate from oxaloacetate by the enzyme phosphoenol pyruvate carboxykinase. That actually, but vertebrates cannot do that. In plants and certain vertebrates and in some microorganisms, they actually can do this. They can actually use the acetate, which they can utilize both as the source of fuel as well as the source of phosphoenol pyruvate to synthesize carbohydrate. Okay, but we vertebrates, we cannot do this. So these organisms, how they are doing this actually? These organisms, they have the cycle, which is called the glyoxylate cycle, and the purpose of the glyoxylate cycle is to produce 4 carbon compounds. For example, that cycle convert the acetate to succinate, which is a 4 carbon compound. And that succinate, if you remember, succinate is one of the intermediate of the TCA cycle. Okay, so the main purpose, we can say, of this cycle, the glyoxylate cycle, is to produce 4 carbon compound, which is actually the intermediate of the TCA cycle. Okay, we'll look at the cycle here. So the cycle starts in the same way as the TCA cycle. Oxaloacetate condensed with acetyl CoA by the same enzyme, citrate synthase, and we get the citrate. And in the next reaction, citrate is converted to isocitrate by the actually 2 consecutive reaction, which is more clearly shown in the TCA cycle. In the next step, this isocitrate undergoes cleavage. Okay, cleavage reaction. It breaks down by the enzyme isocitrate lyase. So already we learned the lyase enzyme. If you remember, lyase's function is to degrade, like break down a compound. So isocitrate lyase breaks down isocitrate, and we get 2 products. One is succinate, another one is glyoxylate. So succinate is actually released from this cycle, but glyoxylate, it remains within the cycle. And succinate is the 4 carbon compound, and we also know that succinate is also intermediate of the TCA cycle. So succinate can easily then enter the TCA cycle. Okay, so that means the organisms which have this cycle, they actually replenishing the TCA cycle by this pathway. And what happened to this glyoxylate? Next, the glyoxylate condensed with another molecule of acetyl CoA by the enzyme malate synthase. Okay, synthase again, condensation, and produce malate, because the enzyme is telling according to the product malate, we get malate, and malate will then undergo dehydrogenation reaction to generate the starting material, which is oxaloacetate. And during that dehydrogenation reaction, a molecule of NADH is produced. Okay, so here the main purpose of this cycle is not the like energy production or even the production of NADH that is actually produced, but the main goal is to produce or provide the 4 carbon compound, which is the intermediate of TCA cycle, so that TCA cycle can run, as well as the intermediates can be utilized to synthesize other biomolecules. Clear?
Yes, ma'am.
Okay, so now we have to look at the relationship between these two cycles, glyoxylate and TCA cycles. So glyoxylate cycles, this cycle occurs in the glyoxysome, which is is a cell organelle, and TCA cycle occurs in mitochondria. But these two cycles, they actually happen simultaneously. And when these cycles are going simultaneously, it is possible for these two cycles to pass the intermediates. Okay, so what happened in this figure, you will see. So how this glyoxylate cycle is happening. So the cell store lipid as triacyl glycerol, because this is the main thing, because we learned that fatty acids is synthesized, fatty acids are the source, or acetyl CoA is the source of glucose. So here we are seeing that fatty acids, which are catabolized into acetyl CoA, which enters the glyoxylate cycle, and we get succinate, and that happens in the glyoxysome. Then succinate goes to the mitochondria and enters the TCA cycle, because we already know succinate is one of the intermediate. Okay, now what is the purpose of this? Here the main job is that the intermediates of the TCA cycle is actually used to synthesize glucose, and that process is called gluconeogenesis. So here if you look at this oxaloacetate, which is the actually ultimate intermediate, from it the cell synthesize glucose, or like hexose is the sugar, ultimately from the hexose sugar other form of the carbohydrates are synthesized, and that reaction occurs in glycolysis, sorry, cytosol. Okay, cytosol, the gluconeogenesis occurs in cytosol. Okay, so now when it will happen? It will happen when there is not enough glucose. Cell don't have glucose, because we already know glucose is the excellent fuel. If glucose is there, the cell will not utilize any other fuel. But in this case, the glucose is not here, but the cell wants to use glucose as the fuel. And for that, cell is synthesizing glucose. And from where? From storage lipid triglycerides. Okay, is it clear?
Yes, ma'am.
Okay, now next question is that, because these two cycles, glyoxylate and TCA cycles, they have like similarities, they have some common enzymes. So that means which cycle will go on, because acetyl CoA is the starting one, which can enter any of these pathway. So which pathway will it go? Okay, so who is going to determine that? And that is determined by the enzyme isocitrate dehydrogenase. Okay, isocitrate dehydrogenase is actually responsible for determining whether isocitrate will be will enter the TCA cycle or the glyoxylate cycle. Okay, Okay, now how is that happening? This enzyme, isocitrate dehydrogenase, it is regulated by phosphorylation and dephosphorylation. When it is phosphorylated, this enzyme is inactivated. So when this enzyme is inactivated, the isocitrate that will enter into the biosynthetic pathway that leads to the glyoxylate cycle, because from the glyoxylate cycle ultimately the goal is to synthesize glucose, that is the biosynthesis. Okay, now when the enzyme is dephosphorylated, the enzyme is activated, then the isocitrate enters the TCA cycle, and then the goal is the production of energy, which is ATP. So in the figure you will see that at first acetyl CoA, that means the preliminary steps are same, condensed with oxaloacetate, and we get a citrate, then eventually citrate is converted to isocitrate, because these two steps are same in both the pathway. Okay, now the question about the isocitrate, whether it is dedicated to glyoxylate cycle or citric acid cycle. So isocitrate here, that is like actually controlled by the active inactivation of the enzyme isocitrate dehydrogenase by phosphorylation and dephosphorylation. So if it is phosphorylated, the enzyme cannot actually catalyze isocitrate, then the isocitrate is lysed by the isocitrate lyase, which generates succinate. Okay, that is the glyoxylate pathway. And if it is activated by dephosphorylation, then isocitrate enters the citric acid cycle, where isocitrate is converted to alpha ketoglutarate, and eventually all the rest of the pathways of the cycle will happen, and ultimately we'll get the reducing power NADH FADH2, ultimately which will then carry the electrons to the oxidative phosphorylation, and that is used to synthesize ATP. Is it clear? Also you can see the other factors are also shown here. Suppose in case of glyoxylate cycle, intermediates of the citric acid cycle, glycolysis, AMP, ADP, they are the inhibitory factors for the glyoxylate cycle, whereas the intermediates of the citric acid cycle, glycolysis, ADP, AMP and ADP, they are actually stimulatory. Okay, stimulatory then, because they are stimulating the phosphatase enzyme, which is actually responsible for the dephosphorylation of the enzyme isocitrate dehydrogenase, whereas they are like inhibitory, that means they inhibits the enzyme kinase, protein kinase. Kinase is the enzyme which adds the phosphate group to its substrate. So if it is inhibited, then the isocitrate dehydrogenase will not be phosphorylated. Clear?
Yes, ma'am.
Okay, so this is the end of our TCA cycle, and TCA cycle is actually a common metabolic pathway for other nutrients as well. That means for fatty acid as well as amino acid. So at first we actually learning the catabolism of glucose, and we have seen how the glucose is catabolized. At first we get pyruvate by glycolysis, then pyruvate is further undergoes oxidative decarboxylation into acetyl CoA. Acetyl CoA enters the TCA cycle, whereas the other nutrients, fatty acid and amino acid, all of them is actually converted to this 2-carbon compound, which is acetyl CoA, and all those acetyl CoA then enter the TCA cycle. Okay, clear?
Yes, ma'am.
Any question? কোনো কোশ্চেন আছে? কোশ্চেন যদি না থাকে তাহলে কি আমরা নেক্সট লেকচার শুরু করব? জাস্ট একটু ইন্ট্রোডাক্টরি দুই একটা স্লাইড হয়তো আমরা গো থ্রু করতে পারি।
নেক্সট লেকচারটা ম্যাম পরের ক্লাসে করি।
জাস্ট দুই একটা স্লাইড, জাস্ট অ্যাবাউট আওয়ার আন্ডারস্ট্যান্ডিং। ওকে, দেখতে পাচ্ছ? Is it visible?
Yes, ma'am.
So the next one we are going to learn catabolism of fatty acid. Fatty acids are the monomer of lipid. So same way we have learned what are the fats of glucose, if you remember. So here we are also focusing on that. The fatty acids actually oxidized, and fatty acids are long hydrocarbon chain. We know that after the oxidation we get acetyl-CoA, and when acetyl-CoA is produced, energy is released. Actually the reducing power is released, which is the NADH. Okay, and we get acetyl-CoA, and this reaction actually the energy which we get from this reaction, that reaction provides 80% of energy in mammalian heart and liver under all physiological circumstances. The electrons which actually released during this oxidation of the long chain fatty acid, those electrons are ultimately transported to the respiratory chain, that is the electron transport chain, which is then utilized to synthesize ATP. Okay, and what happened to the acetyl-CoA? The acetyl-CoA, which is then entering the TCA cycle, which we have already learned what is the result of the TCA cycle. Okay, now in liver, the acetyl-CoA, that means there is enough acetyl-CoA, the TCA cycle is going on and still there is enough acetyl-CoA. The acetyl-CoA is converted to ketone bodies. Ketone bodies are water soluble molecules, or we can also say they are fuels. Okay, they can also be utilized as fuels, which are exported to the brain and other tissue, specifically when glucose is not available, because glucose is the ultimate excellent fuel. But if the glucose is not there, the living organisms, they find another source, and in this case, when there is enough fatty acid which are oxidized and there are enough acetyl-CoA is present, then the liver can actually synthesize ketone bodies from acetyl-CoA and transport those ketone bodies to other tissues or organs, specifically brain. Now what about the plants? In plants actually, because they can synthesize glucose by themselves, so they don't need to depend on fatty acid, which will be providing the fuel. So rather they actually use acetyl-CoA as the biosynthetic precursor than the fuel. Okay, so the pathway by which the fatty acids are oxidized into acetyl-CoA, and that oxidation occurred by a repetitive 4-step reaction or process, and that is called beta oxidation. That means from a fatty acid we get acetyl-CoA. So to produce one molecule of acetyl-CoA from a fatty acid, there is a 4-step reactions. 4-step reactions. So fatty acid is like made up of many carbons, and acetyl-CoA is made up of only 2 carbons. So many acetyl-CoA are produced, and to do that it undergoes repetitive 4-step reactions, and that is called beta oxidation. Okay, so the very first step of the fatty acid catabolism is the beta oxidation, where the long chain fatty acid is converted to 2 carbon compound acetyl CoA. Whereas in case of glucose, what it is? Can anyone tell me? গ্লুকোজের ক্ষেত্রে কি? হ্যালো, ঘুমায় গেছে সব নাকি? গ্লুকোজের ক্ষেত্রে কি? হ্যালো, ক্যান ইউ হিয়ার মি?
জি ম্যাম, শোনা যাচ্ছে।
তো অ্যান্সার করছো না কেন? বলো, গ্লুকোজের ক্ষেত্রে কি? ফ্যাটি অ্যাসিডের ক্ষেত্রে বললাম কি বিটা অক্সিডেশনের দ্বারা আমরা ফ্যাটি অ্যাসিড থেকে অ্যাসিটাইল কোয়ে পাই। আর গ্লুকোজের ক্ষেত্রে কি?
ম্যাম
মিস
গ্লুকোজ থেকে
অক্সিডেশন
গ্লাইকোলাইসিসের মাধ্যমে পাইরুভেট আর পাইরুভেট থেকে অ্যাসিটাইল কোয়ে।
ইয়েস, এইটুক জিনিস সবারই তো বলতে পারার কথা। এইটুক জিনিস যদি তোমরা ধরতে না পারো তাহলে তো তোমরা মেটাবলিজম কি বুঝবা? That is the real thing এখানে happening, and rest of the cycles are same for all the nutrients. তার মানে আমরা এখানে অ্যাসিটাইল কোয়ে পেয়ে গেলাম। That acetyl CoA is in the next step, it will enter the TCA cycle, and the TCA cycle we have learned in the previous lecture. And after the TCA cycle, all the electrons which are carried by the electron carrier NADH, FADH2, that will be transported to the electron transport chain. That is also same. So আমরা যদি একটু মনোযোগ দিয়ে the first step catabolism of that particular nutrient যদি একটু শিখি, the remaining two steps are actually the same for all the nutrients. Okay, now what are the sources of fatty acids from where we can get the fatty acids? So actually there are three sources. We can get the fatty acids from our diet. Same way we learn in case of glucose, if you remember, the feeder pathways from where we get the carbohydrate, from dietary carbohydrate, storage carbohydrate. Okay, that's the same thing here. We can get the fat from our diet, or the fat can be stored within our cells as the lipid droplets, or fats can be synthesized by one organ and then it is exported to another. ওকে, এটা আমরা হয়তো বা দেখিনি আগের ক্ষেত্রে, বাট সেখানেও আমরা দেখেছি যে glucose is synthesized from non-carbohydrate source, gluconeogenesis. We can compare with this third process with that one. Now it is about the organism, specifically the species. They can utilize the three sources under various circumstances, or maybe one or two of them. যেহেতু আমরা we are talking about the metabolism of vertebrates, they actually obtain or get the fatty acids from all the three sources. They get the fat from their diet, mobilize fat, store it in specialized tissue which is called adipose tissue. Adipose tissue is made up of adipocytes, the fat cells, and the liver, which actually can convert the carbohydrate, that is if we have enough carbohydrate, to fat, and then the fat is exported to other tissues. So এই ক্ষেত্রে আর কি যদি আমরা একই ভাবে গ্লুকোজের সাথে কম্পেয়ার করি, glucose we can get, or like what is the source of carbohydrate we are getting from diet, carbohydrate which are stored in our cell, then we are also synthesizing carbohydrate from non-carbohydrate sources. So same thing, that is also the gluconeogenesis occurs in liver of vertebrates. Okay, and triacylglycerols, they actually give us half the energy requirements of some organs, particularly liver, heart, and resting skeletal muscle. Because already আমরা জেনেছি why fatty acids are like very important, or why the living organisms store fat as the source of energy rather than carbohydrate. So there is a some feature of the fatty acid, if you remember. Okay, so the very first one is the dietary source of lipid. In our diet, if we have fat, then that will be digested in our digestive tract. If you learn or completed the GB 203 course, I think you have complete idea about that. But before that process, some term we should know. One is apolipoproteins. These are lipid binding proteins which are like present in blood, and their function is to transport different lipid molecules such as triacylglycerol, phospholipids, cholesterol esters in between organs. Another one, chylomicrons. Chylomicrons are actually made up of triacylglycerols together with cholesterol, dietary cholesterol, and lipoproteins. Together, they give rise to the complex or aggregates called chylomicrons. Okay, now what happens once you have fat in your diet? That will go from your stomach to intestine, small intestine. And when there is fat in the small intestine, the gallbladder will release bile, and in the bile there are bile salts that emulsify the fat, and the fat is emulsified, forming mixed micelles. Then again, in the small intestine, there is this enzyme, intestinal lipase, the lipase enzyme, which degrades the triacylglycerol, the macromolecule triacylglycerol. Once it is degraded, we will get fatty acids. Now, in the next step, the fatty acids and other smaller molecules, they are actually absorbed by the intestinal mucosa. And within the intestinal mucosa, these fatty acids again converted to triacylglycerol. Then these triacylglycerols, they are actually complexed with cholesterol and apolipoproteins and make the aggregate which is called chylomicron. And these chylomicrons then actually travel through the lymphatic system and bloodstream to different tissues. Okay, now in the blood circulation system, which is like the capillary, in the capillary there is again the enzyme which is called lipoprotein lipase, and this lipoprotein lipase is activated by the ApoC2 protein. It activates the lipoprotein lipase when there is the presence of the chylomicron, and they will actually degrade the triacylglycerol into fatty acids and glycerol. So from this capillary, the fatty acids, they will be transported to the different cells. So it depends on the cell, such as if it is myocyte, which is a muscle cell, the fatty acids are utilized to generate energy, and which is used to contract the muscle. Or if it is adipocyte, which is the fat cell, the fatty acids are again esterified to generate triacylglycerol for storage. Okay, is it clear?
Yes, ma'am.
So it is I don't know whoever has already completed GB 203, that it is easy for you to understand. But the thing is that you should complete 201 first and then go for 203. Okay, so let us stop here for today, and we can like go forward from the next lecture in the next class. If it is in person, then we will see each other in the classroom. And if it is not, then we have to do online class. But by that time, hopefully, I will be able to let you know what is our like assignment. I think that is more important than we can conduct presentation, because presentation will be difficult to conduct. If we have to do, then we have to do in a group. Okay, then. Allah Hafiz.
মিস আসসালামু আলাইকুম।
ম্যাম আসসালামু
Assalamu
আলাইকুম ম্যাম।
alaikum.
আসসালামু আলাইকুম।